Uh, okay, I think we're all in now. So I would like to say, on behalf of CanCOVID, bienvenue and welcome. Thank you for coming to today's speaker series. We have with us Dr. Fiona Brinkman, who's here to report on the Genome Canada CANCOGEN project. I'm excited about that. I would like to remind you about some simple Zoom etiquette to please keep your microphones off during the presentation. After Dr. Brinkman speaks, we will have time for questions. You can either raise your hand and ask your question aloud, or you can type your questions in the chat box and I will read them. N'hésitez pas à poser des questions en français si vous préférez. It is my great pleasure to welcome Dr. Fiona Brinkman. Dr. Fiona Brinkman, please take the screen sharing floor.
Okay, thank you. Hello, bonjour. It's my pleasure to provide an overview of this new Canadian COVID-19 Genomics Network, referred to as CANCOGEN or CANCOGEN en français. So first I'm going to start with just doing a little bit of an overview. Oops, sorry, 1 second. This initiative has been possible as part of as a result of a very appreciated $40 million federal investment, with 20 million going to viral sequencing and 20 million going to human sequencing from COVID-19 patients. The goal of this project really is primarily coordinate and fund SARS-CoV-2 and human host genome sequencing efforts, SARS-CoV-2 being the virus that causes COVID-19 disease, up to 150,000 viral sequences and 10,000 human sequences. We basically are going to be integrating this with associated clinical and epidemiological data, otherwise sometimes referred to as metadata in genomics circles. We are going to focus also on facilitating this data sharing more nationally, because as you know, healthcare is a provincial jurisdiction, and also be working on capacity building, including for future outbreak and pandemic preparedness. So this initiative, I have to acknowledge, is really a result of the leadership of several individuals. Genome Canada in particular really stepped up to the plate after the UK started a similar initiative and said, should we start this as well? And there was basically a strong enthusiasm for this to fill some gaps that are critically needed to enable more genomic-based pandemic investigations. So we also have a sort of structure formed with a human sequencing under the an implementation committee headed by Naveed Aziz, and a implementation committee for the viral sequencing headed by Terry Snutch, and a whole range of diverse groups of participants, including a consortium of national and provincial public health labs. With on the viral side, we have Gary Van Domsler of the National Microbiology Labs playing a key role, along with Will Sow on the provincial labs, spearheading, filling in gaps in our metadata organization. We've got hospitals, universities, research institutes, other research institutes, large-scale genome sequencing project centers, and industry are also playing key roles in the support of this Genome Canada organized initiative. Really, though, why are we doing this? So primarily, this is about coordination. So more coordination of viral sequencing and associated clinical and epidemiological data, collection between provinces, in particular, because as I mentioned, healthcare being a provincial jurisdiction, this has led to a bit of a siloing of data and collection, and we need to basically coordinate more across provinces. We can also capitalize on Canadian leadership in genomics, studying the role of human genetics in the outcome of COVID-19 disease. Notably, we can specifically tailor this to Canadians and our genetics and metadata rather than depending on studies spearheaded by other countries. So this is very much focused on Canadian data. However, we're going to align this with global sequencing efforts, including these recently announced national efforts like COG-UK in the UK and SPEARS in the USA. This also, a critical component of this is capacity building, both now and for the future. But really, why are we doing this? So I want to talk more about the benefits of this data and what it provides. So essentially, this is really about looking at transmission when it comes to sequencing SARS-CoV-2 genomes. So there are a number of other benefits that I will go into, but I would say this is one that's being very actively used right now. So basically, we can confirm or transmission trends, capitalizing on these lineages of the virus that form, and looking not just internationally at transmission, but also nationally, provincially, and locally as well. So here's the first virus sequence that was collected from an individual in Wuhan, China. You can see here, I've just got a little purple dot here noting this. Note that it isn't very big genome. This is 30,000 nucleotides. Our human genome is about 3 billion base pairs. But this S here denotes the spike protein, which encodes this protein that you guys will be very familiar with, having seen lots of pictures of this coronavirus floating around everywhere. And so this spike protein is a key component because it binds the human ACE2 receptor in lungs and is basically a key target for vaccine development, etc. So over time, this virus basically can transmit through the population and mutate. This is an isolate collected in Germany in late January that acquired 2 mutations relative to this original Wuhan isolate that was identified in December. In general, we see sort of changes occurring roughly about 1 mutation every 2 weeks. And we can use these mutations basically to start making a family tree of sorts of the relatedness between the different isolates. So here's another Canadian isolate. And so over time, this virus spreads and diversifies, and the location information along with other metadata can help really inform the development of this outbreak. For example, you can see in this huge tree here of isolates collected globally, you can see, so this is just showing here this German isolate, and then it potentially some European isolates being a source of infection for humans in Canada. But as you can see here, this is sort of colored. Actually, I'll show you this picture, which is a bit better. So you can see here, this is a global map of these proposed transmissions mapped out using this genome sequence, literally like a sort of digital code of relatedness between isolates from different individuals. And so we can see here, for example, you get all these isolates in Wuhan and in China that show up here at the base of this tree, and then you get these resulting lineages that form. And you can see here, for example, just even visually, how there's a bunch of isolates at, in Europe, and then many of the isolates in North America, denoted by red here. A significant number of them in this tree are of European origin. There are definitely some of Asian origin as well. But you can see here, for example, in this example of studies of transmission, how these sequences are really helpful to sort of see how transmission was occurring, and in some cases was very critical to identify these sort of similarities that would confirm what was seen with contact tracing, for example. So for example, this is changing the colors here. Here you can see that we have introductions of Canadian isolates that are shown here in orange into Canada from global sources, but predominantly from the US. You can see many of these are associated with these US ones that are in yellow. And then 2 US isolates are showing up here. I just have to change my screen here a little bit so I can see them myself. So these 2 USA isolates are showing up here that are interleaved in some Canadian isolates, showing that we gave back, so to speak, to the US, and they got some isolates from us as well. And needless to say, this has supported, closing our border right now. So if you look here, for example, in BC, you'll see that there's many isolates that will be have been introduced basically from other locations. Basically, international travel was a big factor in precipitating many introductions multiple times in BC. And what was really key is that being able to confirm if this matched travel history. So for example, you might see something where somebody's isolate came from the US, and this made sense in light of where they had traveled. But for example, there's one case that's public about Iran, a person who traveled from Iran, but their isolate did not match an Iran origin. And so this called into question if they'd gotten it from elsewhere. And in Seattle, of course, we had a big issue with isolates showing up being, what's the word I'm looking for? There was a bunch of isolates that basically were showing up in Seattle, and then another isolate came in that basically implied that was related to much earlier isolates from weeks before, which was key to imply that these were not, that there was isolates circulating hidden in this community, and there was basically community transmission going on. So as a result of these kinds of analyses, we can look at transmission trends. We can now look at this regionally, but also what's going to be very important as we start to de-escalate control measures, start to open borders, being able to track and see if isolates come in, what the genome sequence tells us in terms of where this may have come from is going to become very important. I have to say, though, just a cautionary note that it's not a be-all and end-all. In the end, you have to integrate this with other data, okay? And so, but it still can be very useful for cluster investigations, i.e. genomic epidemiology, or noting additional cases that show up, clusters of cases that show up that indicate potentially a new outbreak of related isolates. Also, there's evolving viral characteristics that can impact things like detection methods. You know, we're obviously very interested in developing serology methods, clinical outcomes, strain severity, how well an isolate can transmit, and also might impact effectiveness of healthcare measures, treatments, and vaccines. So for example, there is this isolate that's gotten a bit of press recently, this strain, or maybe I'll call it a lineage, it's not really a strain, but this lineage that's this mutation that has occurred in the spike protein, and there's been concerns that if you look at the G form of this versus the D form at position 614 in the spike protein, you'll see that there was some, D form was predominating, and then the G form became more predominant. So there's concern that maybe this might be a more transmitted version, a transmissible version, and may have differences in terms of its virulence. However, we need to factor in things like the founder effect as illustrated here. So if you have a bunch of different isolates, or a bunch of different lineages in a population, and somebody happens to transmit one over to another population, and this becomes for various reasons, maybe they attended a conference which led to some sort of super spreading events. For example, as you probably heard, there's been choirs and other events that have led to spreading, that you can have all of a sudden one lineage sort of mushroom into a big outbreak as a result of that. And that has nothing to do with its transmissibility. It's a bit of a sort of a chance thing, and what's happening in terms of other data, not the actual virus sequence itself. So we do need to look at integrate this genomic data with other epidemiological data to investigate these things. And also, I have to say, what's going to be really critical is investigating this in the context of how things are changing as we go along. So we need to continue this genome sequencing so we can track how these changes are occurring. This is particularly important for informing things like vaccine components, we need to know early if there's going to be a change in the spike protein, this is shown here, that is going to impact a potential vaccine that's being developed. So this is just showing changes that are occurring, and there are some parts of this protein that are changing rapidly and other parts that are not changing much at all. And so we need to keep track of that. So, so this project, I've focused a lot on talking about the viral side, this CANCOGeN V, as we call it. We've made operating principles for this. We've come up with a sort of a general structure for how this has worked. This has involved a huge effort, actually, just coming up with something that we can get agreement for with public health labs, sequencing centers. We've got to get others on board, making sure we're dealing with ethical, legal, social issues. And then the idea is we're creating a national database, and all of this data will be put out publicly into GSED and other sort of public resources for viral genomic data sharing. And this is certainly, we're partnering again with things like UK, COG UK and other initiatives, not just for the genomic data, but also for this other data, the epidemiological clinical data, which is also really important to get organized. So for the human side, I'm not going to spend as much time on this because we don't have a lot of time. And I think it'd be great if you guys could hear from them some other time in the future also. But I will say that for the human side, this is really becoming important as we see COVID-19 displaying significant inter-individual clinical variability, ranging from very silent infection to lethal disease. We can get these rare unexplained resistance to infections and these rare severe disease, for example, in the very young. And we need to characterize the role of human genetics in this. We know it's a mixed, there's a bunch of different factors that play a role, both environment, genetics, but we need to characterize this better. And we can also potentially deliver new biomarkers that predict people that are at higher risk, highest risk, and implement appropriate healthcare and development of potentially precision therapies, customized more for people depending on their genetic background. Studies, this study is based on a sort of a project-based approach. For the viral side, we are very much focused on trying to sequence as much as we can, in particular dealing with backlog of sequencing, but also sequencing in the future in real time to be able to analyze more rapidly what is going on and how this virus is evolving. In the human side, this is basically based on projects that get research ethics board consent. There's biospecimen data collection, then sequencing, then the data is returned to the PI with controlled access for future studies. So there's a whole infrastructure that's been set up for that. Naveed Aziz was, who chairs this, was I'm very happy he was able to share this slide that, just for reference, I'm not going to go into it much, but just to show you that they are really engaging a bunch of study partners. This is open to all and really aiming to build a national network, and want to hear from projects that basically have particular ideas for where there would be a study that would suit human genome sequencing. Okay, in closing, my last slide, I'd just like to sort of summarize the current activities. We've had some initial fund allocation occurring, focusing on, for the viral side, we've got sort of for sequencing primarily. We've got a lot of people actually just sort of working for free right now, trying to get this going, and we'll sort of backfill in some of this as we go along. But a key component also is just setting up these operating principles and MOUs, building the sequencing and operational infrastructure, and in parallel, sort of working on this protocol standardization, this huge effort to basically coordinate metadata collection, not just here, but also in these other initiatives internationally, and then really ramp up the sequencing, okay? The model for this is, I can't emphasize enough, is this is forming a base for something more. We don't want to just stop here. So we want to basically be able to do this genome sequencing as a model for forming something like a Canadian Pathogen Genomics Network. This is forming a base that we can use for future pandemic preparedness, future outbreak investigations, and not just for human pathogens. We can, obviously there's some components of this that are very much geared for human pathogens, but you know, coronaviruses occur in, that are a problem in pigs, for example, and there's a lot of different pathogens affecting the agri-foods industry, different pathogens affecting forestry, and aspects of what we're setting up here for both pathogen and host we could use for these other organizations. So this is really also a model for data sharing in general. And so I encourage those who are interested in data and data sharing and any aspects of this to contact us if you want to get involved. We would, this is the stage where we really are reaching out to people and wanting to reach out to this CanCOVID initiative as well. If you want to get involved, we've got an email address there, info@genomecanada.ca, that you can use basically to initiate connections. So if you have any questions, I'd be happy to hear from them. Thank you very much. And I just want to acknowledge the great team of people that have been involved in this. They really are working tirelessly. So some of them that'll be on the call, thank you.
Wonderful, thank you so much. And so now we can take questions. Peter Rogan actually asks right away, can you indicate who to contact regarding the CANCOGEN human project? Is that still the info at email address that's right there on the screen?
I think you should just email that, but Naveed Aziz is definitely the lead on that side of the project, and can certainly provide that email. But I think if you go to email at genomecanada.ca, they'll be very responsive in providing that.
Great. And Ali asks, Dr. Brinkman, if researchers are interested in having access to anonymous COVID-19 patients, what resource would you recommend? Patients' information.
That is difficult for me to answer. I have to say, in terms of the sequencing is being released, at least, you know, when you say information, there's a lot of different kinds of information. So for the viral sequencing, we are putting all of that out onto the to GSED and other initiatives where this data will be made available with some minimal metadata. We have to be very careful about what kinds of data are released with these to avoid identifiability issues. But for the human side, there's an infrastructure for what can be shared and what can't be shared. I think really the best thing is to get in touch with the different groups. But if you're particularly interested in viral sequence data, through this initiative, we will really, through GSED is the way to get that sequence. And if that's what you're referring to, or is there another aspect?
Okay, we'll see if we get a follow up on that.
Okay.
Oh yes, confirming. Yes, that was
okay.
That was the
okay.
And so Moto asks, how can we get samples for sequencing in our communities?
Yes, so you mean samples that would be actually that you'd want to do, oh, you just want to do your own sequencing? Okay, if somebody is interested in doing sequencing, they can definitely contact us, but we do have an infrastructure set up to do sequencing associated with each province. But in short, I would ask them to get in touch with us with their particular interest.
Okay, great. Then there's a warning, beware of GISAID terms and conditions of data usage.
Yes, we are very aware of that. And so we are very interested in FAIR principles in terms of providing all the data with that support FAIR principles. But yes, we are very aware of that. And in short, are interested in setting up infrastructure. One of the challenges we've had here in Canada is that we don't really have our own NCBI. We don't have our own infrastructure. So hopefully as part of this, we can generate some infrastructure that will help. But I have to say that this is sort of a staged approach, that we're going to be first, we're building this in the air, so to speak. We're basically building this plane in the air, as I think has been mentioned before. But I have to say, this is not something that's going to be, we're going to be sort of just trying to do what we can to address this current pandemic very rapidly and getting this sequence out the door as quickly as possible. There'll be, as we evolve though, and develop this infrastructure more, we can be a little bit more sophisticated, I guess, in how we're releasing data.
Mm-hmm. Peter Rogan, did you have your hand up? Yes.
We can't hear you.
Oh, yeah. Oh, unfortunately, I'm so sorry. We can't hear you at all. No.
Sorry.
It sounds like a chipmunk talking, so I don't think that's right.
Yeah.
Did we have any other questions while Peter's trying to work out his sound issues?
I will just add a commentary that regarding that issue of the, some of the things that will come out, in the news, for example, that there's a new isolate or strain or lineage that is more transmissible. Just want to add a cautionary note to that we do really need that other epidemiological data, that really this is one piece in a puzzle of collective data that needs to be collectively looked at. But this genome sequence data is going to be absolutely critical in terms of, as we move forward, tracking how things are changing. And what's nice about it is it's really digital. It's very definitive. You either get the genome sequence or you don't, in most cases.
I like definitive.
More questions there.
Yes. Are you considering other types of sequencing, RNA sequence, DRIP sequence, CHIP, understand the mechanism of the host-virus interaction?
Yeah, so that would be part of these clinical studies. That is certainly on the viral side. We are very much focused on the viral sequencing and getting the data out and in an organized fashion, in a coordinated fashion. The human side is certainly a little bit more study-centered, right, around certain research questions. And so in that case, absolutely, there would be, I would assume in some studies, I don't know the details of all the different ones being proposed, that you would be looking at that further. However, really, I think what it comes down to is there is a lot of data being generated about the coronavirus, and there's other initiatives that are also looking at other aspects, looking at gene expression, looking at serology, etc. And we need to integrate those. So we can hopefully really use this pandemic. It's awful this pandemic has occurred, but this is really spurring on some innovation in this area. So I think we can really use this very positively to facilitate more interactions between different groups. And
Absolutely.
I see there's another.
Yeah, so Brian Corey says, if anyone is sequencing human immune response to COVID-19 and have public data, we would be interested in talking to you. Support at ireceptor.org. So if anyone here. The Darius comments that the graph for the UK showed only one type compared to other countries. What is the reason? So it only showed, I believe D was the bottom and G was the top, and it only had D. So it didn't show the second.
Sorry, which slide was that? I'm on my slides now. Should I be sharing them again?
Sure.
I can share my screen again, just because I know we're almost out of time, but I think we can just do this. So was it on this slide, or I'm just gonna open up my chat?
I think it's the one with all of the countries, all the different graphs of each country.
Okay, just a minute. This one, maybe?
Go further. Where the countries were charted individually with the two different types. And it had North America and Europe.
This one maybe? Or, sorry, if somebody, I'm just trying to look to see if there's a copy here. Graph for UK. Oh, is it maybe just that you don't have Darius? Are you talking about a different graph than I've shown?
The bar graphs that are colored blue and orange. Not that one, not that one. Keep going.
I'm not sure about, I don't have a bar graph.
Yeah, the D versus G.
Oh, yes. This
Yes,
one.
that one.
Yes, okay, thank you. Sorry for that. So basically you were asking about how it only showed one type. Yeah, so basically that's the good question. This is what led to this interest in, is this sort of blue form, shall we say, lineage, is it more transmissible? And that is one reason it took off in England. But I actually think you have to be very careful to look at this, because we had cases come in, for example, we had the G form and the D form present both in Seattle, and it's not like so far one has overtaken the other, at least at the time when I looked at the data. Whereas in England, it just might be just chance. This is that founder effect, that if this case here was somebody who came over to England, and then there was a few super spreader events associated with that case, then that could just take off. So it's really a little bit of a chance effect. You know, does the virus literally get lucky with going into somebody who's moving around a lot, or going to you know, a buffet or something, and transmits significantly? So this is where getting that genomic data with the other data is really key. But this is very critical that we look at these, and we have to keep tracking whether there's going to be some other changes that occur that are going to have an impact, particularly for vaccine development.
That's great. Thank you so much. And thank you everyone for joining us, and thank you for all of your great questions. And thank you, Dr. Brinkman. Have a good afternoon, everyone.